Hello, my name is Riley Gillis and I am a research fellow in Dr. Lee's laboratory. If you're watching this video, you are likely working with Candida auris and or performing XTT assays. So this is meant to be kind of an extended protocol and explanation so that you can get comfortable with this assay and working with this type of yeast. Now throughout this video, you will also see Hannah Schultz. She is a medical student that is assisting me. And we are working right now to test various compounds with antifungal effects on Candida auris, as it is an emerging pathogen. Now, as you may know, Candida auris is a bit more nefarious than its other Candida species counterparts like Candida albicans. It is worked with under enhanced BSL-2 precautions, and during infection, it kills about 30 to 70% of people. Some people who are colonized for life, and so it is a very tricky pathogen. It colonizes surfaces super easily. It's resistant to many different classes of antifungals, so there's three major ones. Fluconazole, about 90% of strains are resistant to that. Amphotericin B is about 30%, and then echinocandins, only about 2% of strains are resistant to that at this point. But the worry is, of course, that will increase in the future due to overprescription of these antifungals. And so the point of the XTT is to, one, test for, basically, is this compound going to be effective against biofilm formation, as that is a major driver of virulence, especially in catheters and other indwelling devices in nosocomial settings and long-term care facilities. And then two would be to test for synergy between these compounds. So briefly, the protocol for an XTT is going to be you are growing the yeast in agar and then in broth, just so you have a bunch of these cells that you can use for your assay. You're going to seed those yeast cells into microtiter plates. Okay, so standard 96 well plates, you're going to seed them in there, let them incubate, so they're going to form biofilms on the bottom of the plate. And then you're going to expose the plates to the compounds in varying concentrations. It's at this point that you can also test for synergy if you want to do a two-dimensional or three-dimensional checkerboard with serial dilutions. And then finally, you will wash the plates just to get all of the media off, and then you're going to add XTT, which is our colorimetric compound. As the yeast cells are more metabolically active, as you lower the concentration of whatever compound you're using, you'll see that those cells are going to turn darker because of this chemical. And so we should see a nice gradient if we're doing serial dilutions that show us, is this compound going to be effective at dispersing, killing, or preventing biofilm formation. After which we will read it in a plate reader and do some stat analysis. So stay tuned. Feel free to play the video at 1.5 times speed. Okay, so to start, you are going to thaw a sample of Candida auris, or whatever Candida species you're working with, from the -80 freezer. This simply consists of taking out of the -80 freezer, putting it in a biosafety cabinet, and letting it partially thaw. You don't want to let it completely thaw because you're not going to be using all of those cells. At that point, you will take a inoculating loop. You'll see there's a large side and a small side. I would use the large side. I'd use the large side first. You can really use either side, to be honest. And then streak a YPD plate after you've made the YPD agar. There are instructions in our notebooks, so just refer to mine and Hannah Schultz notebooks. You're going to do a streaking dilution method where you grab a loopful from the vial with your sterile, individually packaged inoculating loop. You're going to streak one quadrant of the plate, flip it over, run from that quadrant to another quadrant, run from that quadrant to another quadrant, run from that quadrant to another quadrant. We aren't using Bunsen burners here. That's why you flip it over after the first streak, so that you get a more dilute streak with each quadrant. And hopefully after you grow that at 37 degrees Celsius, for a day or two, you will have isolated colonies. They will appear beige, slightly raised, very round, small, quasi E. coli. Okay, after you have streaked your plate, you can take an isolated colony. So you have your plate, you have some colonies, right? You're gonna take an isolated colony with another loop, you know, a day after, whatever. You're just gonna grab that colony. You can grab a couple if you want. Just make sure that you're actually grabbing auris colonies and not something else that's contamination. It should be pretty obvious if it's not an auris colony. So you'll take that and then you will simply put it into a broth culture, mix it around. It'll be a 50 milliliter conical with maybe about 10 milliliters of fluid. And then you'll keep that shaking at 37 degrees Celsius under CO2. Okay, the next step in our assay is going to be to enumerate the cells. And so you'll have an undisclosed amount of cells that have grown in this broth culture overnight. And we want to make sure that when we seed our plates, that there's the exact same concentration of cells across all of the different plates that we do, so that we're comparing apples to apples and not apples to oranges when we're actually comparing the effects of these antifungal agents. And so the way that we're going to do this is you're going to centrifuge that conical. You're going to wash it a couple times with PBS. We'll have a video on that. It essentially consists of decanting and then adding PBS. And then you're going to resuspend it in about 20 to 40 milliliters of RPMI 1640 with L-glutamine, buffered with 165 millimolar MOPS. You'll find instructions on this in our laboratory notebooks. So yeah, just resuspend it in about that much. Then you're going to enumerate via Hemocytometer microscopy. That's a fancy word that just says counting cells on a grid. And so we have these microscope slides that you're going to prepare a dilution in, put them in. You're going to count in quadrants. And then after which you will do some math and you'll actually be able to quantify how many cells are in your culture. You'll be able to make a dilution so that you have the same amount of cells. The desired dilution for Candida species are 1 times 10 to the 6th cells per milliliter. So then after that point you can seed your plates and incubate the biofilms. Right now we have woken up some cells from -80. This is Candida albicans that we're working with at BSL2 currently. And then we've got those started on YPD Agar plates. We've taken an isolated colony and inoculated a YPD broth culture with that overnight. And then this morning, all that we are doing is we are washing the pellet. So we're spinning down our broth culture and trying to get all of the YPD agar out so that it's just a clean pellet of just cells. And so what we've done is we've spun it down. So you'll see here. So this is our culture. Originally there was broth in here. We've spun it down, and so you can see there's a pellet right there of cells. It's pretty tightly packed. We did 3,000 Gs for 10 minutes. And then on top is PBS. That's what we're washing with, phosphate buffered saline. And so this step simply consists of centrifugation and then decanting twice. And then we're going to resuspend it in RPMI 1640 media and plate it in our plates at the desired concentration, which is 1 times 10 to the 6th cells per milliliter. So here I just have PBS. I'm going to grab a little bit more just because I like to do about 25 milliliters per wash. You could do less, you could do more, it doesn't really matter. So following good aseptic technique, we're double gloved, and also we've taped for a seal on our lab coats. If you're doing this with auris, then you'll want to have a disposable lab coat on as well, and bleach inside the hood. So I'm just gonna pour some, avoid touching the tip to anything. All right, we're good, because if you touch the threads, then you kind of contaminate it. And you notice I tilted it, you don't want to be hovering over anything. All right, so I got about 25 milliliters. Whenever we're spinning this down, we want to parafilm the top to create a seal. Otherwise, and in fact, you may also just want on top of this a rotor that seals to avoid colonization of your laboratory. So this is a tightly packed pellet. I'm just going to decant. No need to use a pipette at this point. Easy peasy. This is into bleach. Just have some bleach at the bottom of this. Recap, that pellet's still intact. And now we're going to want to resuspend the pellet in PBS and wash it one more time. This is our last wash, so just gonna pour it. A little shaky today, but it's okay. All right. So we're actually all done with this PBS, so I'm just going to throw it in here. You want to vent it in the bag so that it doesn't pop off in the autoclave and explode. All right, so at this point you can vortex vigorously, or I prefer to use a serological pipette to resuspend. I think it's safer. It creates less aerosols, or fewer, I guess. I'm using a 25 milliliter pipette so that I don't mistakenly pull up too much, because there's only 25, and contaminate the pipette. All right, now all that I'm going to do is resuspend vigorously via aspiration and dispensing. You'll see it's kind of breaking up. There are little specks. You'll want to do this until it's completely homogeneous. All right, and now what I'm going to do is I'm going to put the tip of the pipette to the side and slowly dispense and see if I see any specks. If I see any specks, I'll mix it a couple more times. Because specks indicate that you have biofilm that was not broken up. But yeah, we look good. So I'm going to cap this 50 milliliter conical. I'm going to re-sleeve this pipette and discard it in our biohazardous waste. Now I'm going to take this and let's see, is this okay, and parafilm the top. We're not using airtight rotors for this, primarily because there's not really a respiratory risk, and so we just have a protocol in case there's a spill inside the centrifuge on how to contain that. All right, you can just stretch it around, seal it. Oops. Good. All right, now because this is BSL2, we would want to use bleach. Right now we're just working with albicans, so we're using ethanol. I'm going to spray it off, and then an assistant is going to take it from you. Thank you. And then when you're coming out of the hood, you want to spray your gloves down and doff using the technique that you learned in your... There we go. Oh. So you don't want to touch your second pair of gloves. That's why we use this technique. All right, you're going to put them in there, come out, and voila. Now we'll centrifuge. All right, so we've just centrifuged. Hannah just centrifuged this at 3,000 Gs for 10 minutes. Acceleration and deceleration of 9. This is our last wash. So this was our second wash with PBS. I'm going to decant into the bleach. You see we still have a nice pellet here. And then you're going to add roughly like somewhere between 20 and 40 milliliters of RPMI to resuspend. It's going to depend on how long of a broth culture you had and how much media you had originally in the broth culture. So obviously, you know, if you have more cells in your pellet, you're going to need more RPMI to dilute it down to the target concentration. But the goal of this step is just to get it kind of in the ballpark so that we can count, and then we'll make a dilution. Oops. So I'm just gonna mix this a little bit. I don't know if anything settles in this. We have it covered with tin foil because it's light sensitive, so make sure to keep it out of the light. So I'm just gonna do about 30. Okay. Reseal this. You'll see I have an Eppendorf in here for making a small aliquot. This is for our cell counting. So we'll enumerate via hemocytometer microscopy, which is just a fancy word for counting cells in a grid with the microscope. So I'm going to make sure this is homogeneous so that I can get an accurate cell count. This is one of the most critical steps to make sure that it's homogeneous. Careful not to pull any into the pipette. You see I'm kind of getting up there. Alright, I don't see any specks, but I'll do it one more time just to make sure. Okay, no specks. I'm just going to take a little bit. You see how there's some left in the tip, and that's going to be our aliquot. You can perform the hemocytometer dilutions in the hood, or you can do it on the bench top. I'm just going to do it on the bench top because it's going to be outside the hood anyways, so might as well. But see, we have a little bit. To re-sleeve this. Going to spray this off, and you can either hand it out to an assistant or you can spray your gloves off and doff like this. It's a little challenging. Alright, coming out of the hood. Alright, now we will make our dilution to count. So we found that for an overnight culture, a 1 to 250 dilution is good for the initial dilution, and then around a 1 to 2 dilution is good for the confirmation count after you make your dilution. So right now we probably have somewhere around 1 times 10 to the 8th cells in there, and we want 1 times 10 to the 6th. So So we're just gonna count these, do some math, make a dilution. So you see I'm mixing it. It's a little harder with small volumes to make sure you don't get those droplets up there and so that you're able to mix it. So you can do it either this way or you can use a micropipetter to mix it. But as long as it's roughly homogeneous. So I'm going to take a P1000. So we're doing a 1 to 250. Do not pipette anything smaller than 1 microliter. Okay, so I'm going to do 2 microliters into 500 because there's actually a 2 microliter mark on the pipettes or on the pipette tips. So I'm going to grab 500 microliters of PBS. That's what we're going to be diluting in. You can do PBS or RP, doesn't matter. Avoid putting the pipette into the stock solutions. So what I'm going to do is tilt it so that I don't actually really have to stick the physical pipette in, only the tip is in there. So you see I got 500. You're going to add your larger volume first. I'm just going to add it to the bottom of this. I'm going to keep this pipette for mixing. Okay. Make sure this is mixed up. Now grab your 2 microliters. Okay, so you'll see it should go to that line. It might be hard to see on the camera. This one is not because these haven't been calibrated. And so you'll want to over pipette until you actually get to that line for an accurate count. So I'm going slightly past the first stop. That's not an ideal method, but and then kind of trial by fire, trial by error. Now you'll see it's at that mark. So I know there's 2 microliters in here because I know that the tips are accurate. All right, so I'm going to take this mixed stuff. I'm going to add it to here. There's going to be some residual cells, so you'll want to mix it with the tip to wash that residual mixture off of the pipette tip, especially with such low volumes. All right, pretty well mixed. Okay, and then you'll take your pipette that you saved from earlier and make sure the mixture is homogeneous. Excellent. Okay, now I'm going to grab the hemocytometer. Alright, so before I add this to the hemocytometer, just going to make sure it's mixed really well. I'm going to add 10 milliliters to each side, 10 microliters, to each side. And so you'll see there's notches in this slide. There's a front and a back. And so you can count both sides or just one side. Today, for time, we're just going to count one side. It would technically be more accurate if you put some in both. But I'm just going to grab 10 microliters of our mixed solution. And you put the cover slip right here. And then just very carefully place the tip in the notch and dispense. Okay, so now that we have this, we're going to take our microscope, set it on the stage, move it to the right side, so there's a front and a back. We're just counting the front right now. Starting at the 4x lens, gonna go up, there it is, change to the 10x. All right, so and you might consider changing the diaphragm and the light really quickly. All right. So we have it at 10x. You can actually also go to 40x, but I'll just show it here. So you're going to count all of the grids, okay, from top left to bottom right. You'll see there's 4 corners with a cross in the middle. Each of the corners contains 16 squares. Okay, you're going to count all of the cells in those. If there's too many, you're going to dilute more. If there's too few, you're going to make another dilution of less. You'll just count them. You'll average all of the corners. So I'll count all of them, divide by 4 after adding them up, and then multiply it by 10 to the 4th in order to get the cells per milliliter. All right, so we have done our math. We got a sum of 343 between the 4 squares divided by 4 times 10 to the 4th times our dilution of 250, and that gives us a final cell density of 2.144 times 10 to the 8th cells per milliliter. And so we're going to use an equation that is C1V1 equals C2V2. C1 and V1, this is your initial concentration and the initial volume that you're working with. And then this is the diluted concentration and the diluted volume that you're working with. And so if we know how much we're starting with in terms of concentration, and we know our target concentration and our target volume, then we can solve for however much of the initial volume that we are going to add to the RPMI to get our desired concentration. And so it's a very, very simple equation. We already did our counting and our math, and we got 2.144 times 10 to the 8th cells per milliliter of our initial concentration. So this is what is in our culture that we've already washed 2 times with PBS and resuspended in 20 to 40 milliliters of RPMI. And this is going to be multiplied by X milliliters. We don't know this yet because this is going to be however much of this we are adding to V2 to get C2. So, and then this is going to equal our target concentration, 1 times 10 to the 6th cells per milliliter times, we're doing 4 plates, and so I'm going to make 40 milliliters because it is 100 microliters per well, 96 wells. We're not using the negative control lane. So it's going to be even less than that, and we want some extra because you're always going to have some of the volume that's lost to, residual liquid that's on the boat or in the tube or whatever. All right, so now to solve for X, we're just going to do X mils equals 1 times 10 to the 6th, 40 times 10 to the 6th, right, because this times this, divided by 2.144 times 10 to the 8th. And if you include the units here, right, these two are going to cancel out, so this is just going to be cells up here. Down here it's going to be cells over milliliters. All right, so these are going to cancel out and you're going to get milliliters on top. All right, so we plugged that into the calculator and we got 18.656 times 10 to the negative second milliliters. We're just going to move the decimal place over twice because of the 10 to the second. And the amount of mils that you're going to get is going to be 0.18656 mils, which is the same as 187 microliters. So in other words, we're going to add 187 microliters of our original cell suspension. This is what we washed twice and then resuspended in a kind of arbitrary amount of RPMI. Okay, so 187 microliters of this into 40 milliliters of RPMI to get 40 milliliters of RPMI that is at 1 times 10 to the 6th cells per milliliter. This is important because we want our plates to be standardized across every single experiment so that we're working with the exact same cell densities so that we can compare apples to apples and not apples to oranges. So hopefully the biofilms will form consistently across every single plate in your experiments, and you'll actually be able to compare test mediums and see, which compounds are more effective at dispersing biofilms. All right, so we're making our dilution to 1 times 10 to the 6th cells. 40 milliliters of that. So I'm going to grab 40 milliliters of RPMI 1640. Into a new conical. I'm going to save this for mixing purposes, so I'm just going to re-sleeve it right now and then we'll use it again here in a sec. Alright, so you said it was 137?
187.
187, okay. So Hannah already set this for me, which is great. With auris, you might want to mix this with a pipette. This is albicans, so I'm not worried about just doing it this way. You could also use a vortex, or just make sure it's very homogeneous. It settles very quickly. Okay. So we're grabbing 137 microliters. Roughly. Or 187. Sorry. Alright, I'm putting it in here. I'm gonna wash a couple times. You can see it mix. I don't know if you saw that, but you could see it go into solution. Alright, get rid of our tip. Close this up. And actually, I'll just do it this way too. But with auris, you'll probably want to use a serological to mix. All right. And I'm just going to grab some of this. Just an aliquot. So I get paranoid that it's not mixed well enough. Alright. Okay. Close this. We're going to make a 1 to 2 dilution and then count again. and then hopefully we're at our target concentration, and then we will seed our plates. All right, so Hannah has done the math and prepared the right suspension of cells, pretty much exactly 1 × 10⁶, so she did a really good job, but it just has to be roughly around there. So we're doing 4 plates, so we made 40 milliliters. We don't need this anymore. So you can either pour it out into the bleach, right? This is the non-diluted. Yeah, okay. So you can either pour this out into the bleach, or it's actually safer, as long as it's not going to spill, to just vent it and autoclave it. And so that's what we'll have you do if you don't have a whole lot of trash. All right. So I'm gonna open a plate. You should have a marker in here too when you do this, but you can do it after as well. You're gonna want to label your plates with your name, the date, what's in them. So Candida albicans or Candida auris, XTT, and then plate number 1. You can also choose to include the compound. So if whatever you're testing it against for the XTT assay. Great. These ones come with lids, and it's just going to be 100 microliters per well. So just organize my stuff. It's getting full. All right. So this is set to 100. We're using sterile tips. I'm gonna make sure this is mixed up really well. Okay, should be good. I'm just gonna pour out about 20 milliliters because I'm just doing 2 plates. The other plates are Hannah's. You can choose to pipette this if you're working with auris. So lanes 1 through 10 are going to be your testing lanes. Lane 11 is going to be your positive control. Lane 12 is going to be your negative control. So we're not going to have anything in lane 12. Make sure when you pipette that it's level across all of them. And then you can set the lid down. And then after you're done, you should dispense your tips. Okay, cover the plate and seal with parafilm, and then incubate at 37 degrees Celsius for however long. Okay, so now our Candida albicans biofilms have been incubating at 37 degrees under CO2 for 24 hours. You can adjust that length based on what the assay is testing for. And so for really mature biofilms, you can go like 96 hours. And then for planktonic cells and active biofilm formation, you can do less than 24 hours. But we're doing 24 to 48 hours. What this step is going to consist of is we're going to aspirate the media that's left over from the homogeneous cell mixture that we seeded into the plates. So we're going to take that out. It's going to be about 80 microliters without disturbing the biofilms at the bottom of the wells. And then we're going to add 300 microliters of PBS and wash 2 to 3 times. We've found that with enhanced BSL2 precautions, this step can take a very long time if you're washing 3 times, because standard XTTs will have you flick the the supernatant out and then slap it onto some towels. But obviously that's not an option for BSL-2 due to the aerosol risk. And so what we'll do is we'll be using a pipette to pull up, dispense into bleach, and then add some more. And then at the conclusion of that, we are going to add 100 microliters of RPMI to every single well except for our negative control, which is going to be in column 12, and our very first column. And the reason that we do that is you're going to have, you know, a bunch of these wells, right, in your 96 well plate. And so we're adding 100, 100, 100, so a negative control over here. You're gonna add 200 of your initial dilution of the compound that you're testing. So in my case, that will be heparin today. Hannah will be doing doxycycline today. You're adding 200 of that right here, and then mixing it up, transferring 100 to this one. So there's going to be 100 of that original mixture plus 100 of the RPMI, and essentially what you have then is a 1-to-2 dilution. So then you're going to mix that up again, transfer another 100. Right, so this is another 1-to-2 dilution. So if we started out at 30 mg per mil concentration, then this one would be 15, this one would be 7.5, and so on. And so this is the gradient that you get, and that's important for the XTT, so you can actually quantify what is the minimum inhibitory concentration of the compound. Okay, so in other words, you know, what is the minimum amount that we need to disturb the biofilm in order for it to be effective, so that we're not over-prescribing or, you know, whatever. And then you'll do that to your last one, besides the positive and negative controls. Sorry, you're not going to add any to the positive control. And then the last 100 microliters is going to go out to the trash. Okay? So that every single well after this is just going to have 100 microliters, right? Because this had 200, we took 100 out, and then this had 200, we took 100 out. So everything's going to have 100 microliters except for the negative control, which will be left empty. Now, then you can incubate it for 24 hours or more based on whatever you're testing. One more thing that I want to draw on the board before we actually get the plates out and show you is going to be tip placement within the plates. And so what you'll see is that for every single well, except for your negative control, there's going to be a biofilm at the bottom of the well. Depending on how long you incubated it for, it may be more apparent or less apparent, but regardless, there's going to be some sort of biofilm formed on the bottom of the well by the yeast. Okay, and so when we're aspirating, washing, and doing our serial dilutions, you do not want to disturb this biofilm. Okay, so when you go in with your tip, that's going to disturb the biofilm, right? We don't want that. What you want to do is kind of go in the corner, okay? So that'll disturb much less of the biofilm, and it'll help you place it at the exact same spot in every single well, so that, you know, you kind of have a constant across all wells where you're touching them, and you're not scraping either. You're just kind of setting it in that corner, and that's what we want. And so it's kind of hard when you have the plate, as you'll see in the hood, if you have it flat on the ground or on the table of the biosafety cabinet. And so my recommendation would not be to cant the tips, it would be to cant the plates and go directly up and down with the tips. So what's that going to look like? That's going to look like you have the plate tilted, okay? And then you come in, right, straight up and down, right? Pull up, add whatever, you know, if you're mixing, whatever you're doing, right? And then take the tip out, move it over to the next well, touch it in the exact same spot right there, right? That's going to make it a lot easier, and I think your results are going to appreciate it as well. Okay, so now we're going to get the plates out of the incubator, and then we'll actually perform our washes and our serial dilutions. Okay, so here we have our plates. We just got them out of the incubator. They're kind of stuck together a little bit. So don't try to unstick them outside of the incubator, right? Because you might actually break the seal, outside of the biosafety cabinet, because you might break the seal and then potentially contaminate your workspace. So you can see there's a little bit of condensation, but you should be able to see there's kind of some turbidity in these wells, right? Whereas the negative controls, when you compare it to the negative controls, there's nothing in the negative controls. And so that's indicative of biofilm growth, however minimal. So we might be able to see the three-dimensional architecture. No, you can't. It's kind of hard to see. But you'll see I have my plates labeled, right? Name, initials, XTT, Candida albicans, and then heparin, and then this is plate 2. Parafilm in the incubators gets it like melts almost, and so it kind of gets sticky. There we go. Okay, we might just cut this part out, but that's okay. Okay, so hopefully you can see it a little bit better now. There's some, residual fluid in those wells. Oops. All right. So first things first, I'm going to open this up and put some PBS in here. Okay, we have our 1X PBS. For more information, you can visit our lab notebooks. Don't do that. All right. This video is sponsored by Reno. Pick up your Reno pipette tips. You'll get them life-changing. All right. Okay, so there should be about 80 microliters of soup that's left in here. And so we have this set to 80. All right. And you'll see what I'm talking about, okay? How it's difficult to see what you're doing with it flat on the ground. And it's not very user-friendly, ergonomics-wise either. And so I just like to pick it up and tilt it, okay? And just be careful not to drop it. Then I'm gonna go straight down and aspirate, okay? So straight down, just get as much as you can. So some of them are gonna have more, some of them are gonna have less just due to evaporation. So get as much as you can. You can see that first, you can see the biofilms there. Look at 12 compared to 1 through 10. Okay, squirt into your bleach. Okay, touching the same spot. Into the bleach, and there's still a little bit left, but that's going to be okay because we're going to wash it. That's the point of the washes. Okay, into the bleach, into the bleach. Especially if you're testing something like ethanol, there's going to be more evaporation in the wells that have a higher concentration of ethanol. And since this since this PBS is actually going to be in this plate and we're not reusing it, I'm actually just going to use the same pipette tips. But I'm going to change it to 300. And it's at this point too that you should document if there's no visible biofilm on some of the wells. For instance, it came off when you were washing, then you'd want to document that and then exclude that from your results. All right, 300. Okay, at this point you will aspirate once more and dispense into your bleach. Then you'll wash that 2 to 3 times per plate. Okay, so we've finished our washes. Now, I'm just going to add 100 microliters of RPMI to every lane except lane 1 and lane 12, our positive control. So this will be what's actually making the dilutions. When we the 1 to 2 dilutions when we serially dilute. So make sure you're at 100. Almost forgot. Okay. We're just doing 100. 1. Okay, so 3. Excellent. Okay, and now we're going to load load my heparin in this case into column 1. All right. So now we are adding our heparin, or whatever compound you're testing. So this is about 3.3x heparin, which is about 3.3 thousand units per milliliter. I'm just mixing it, making sure it's homogeneous. And you're going to put 200 into the first column. A, B. Okay, now we are ready to do our serial dilutions. I'm going to set this just out of the way. I'm going to grab this, and it's going to be hard to go this way. Just from a visual perspective, the reflection of the glass and the plates and whatever. Right, so I'm actually going to go like this. Let me get a better grip on that. There we go. Okay. So this has already been mixed. So just grab 100. 1, 2, 3, 4, 5, 6, 7. That was a 1 to 2 dilution. 1, 2, 3, 4, 5, 6, 7. It's another 1 to 2 dilution. 1, 2, 3, 4, 5, 6, 7. So I'm touching the same corners of each plate. 4, 5, 6, 7. Excuse me, same corners of each well. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. Okay, and I'm not going to add any to 11 because that is our positive control. So I'm just going to trash the last 100 microliters. You'll see, well, it's hard to see on that side, but there's 100 microliters per well except for lane 12, which is our negative control. Okay? So now you will cover and parafilm. I don't know, was it back here? Thank you. Okay. And at this point, after you've covered it, you would be good to spray it off, hand it out to your assistant, do your other plates or whatever. Doff your gloves and come out of the hood. Uh-oh. It's okay, we'll just cut this. Alright folks, so we are back after our 24-hour incubation with our serially diluted compound. So in this case for me it's heparin. There should be a gradient of inhibition. It's probably going to be hard to see before we add the colorimetric XTT. But I'm just going to remove about 100 microliters to wash, and then I'm going to wash about, well, so you can do 3 times, or in this case we're just going to be doing 1 time because we're doing enhanced BSL-2 precautions, and so we found that, you know, it's going to take a pretty crazy amount of time if you're aspirating with the pipette instead of tapping it out like you would in a regular assay. So if you're doing it at BSL-1 and you're able to flick it out and tap it out instead of doing what I'm doing right now, then 3 times would be preferable, but for time and safety's sake, we're just washing once. And then Hannah will add 100 microliters of XTT. And then we'll incubate for 2 hours. And then transfer the Supernatant to a new plate and read it in the plate reader. So pretty simple. I'm just gonna go ahead and actually I'm gonna switch my tips because Hannah's working with doxycycline, and so I don't want to contaminate this PBS with my heparin. We are going to add XTT to column 12. So don't forget that. XTT gets added to everything. You'll notice depending on what you eat, you might be more or less shaky. I just had a protein bar this morning, so I found that for me personally, if I eat breakfast, I'm less shaky, but some people are the opposite. Okay, so now we're making our XTT after we've washed our plates. XTT is light sensitive, So all that I'm going to do is we've made 10 milliliter aliquots and frozen them at -80. I'm going to combine all of them. Is that all of it? Okay, that's roughly all of it. And then add 1 microliter per plate of menadione, which is fungistatic and also extremely hazardous. So you want to be careful working with that. If we have to go in for more, we can get that residual stuff out. Hopefully this will be enough though. Okay. This is our menadione. It's just right there. I'm just going to mix it a little bit. Nothing too crazy. It should remain in solution though. Okay. Now we're going to take 4 microliters of this since we're doing 4 plates between Hannah and I. This evaporates quickly because it's dissolved in acetone, and so you want to be pretty quick about it. And I would wash a couple times once you're in there. Okay, should be good.
Okay. So now that we've washed our plates, we are going to add our XTT menadione solution, which is our colorimetric part of our assay. It is light sensitive, so I just poured it from this tinfoil covered tube into here. So we want to be a little bit expedient about this, but we're going to add it to all 12 of our columns. So we want it in every single one, including our negative control. And we are going to add 100 microliters to all of them. Now that we've got it in all of our wells, I'm going to cover this plate and we're going to put it back in the incubator for 2 to 3 hours at 37 degrees, and then we'll take it back out and put it in our plate reader.
Okay, so now we've done our 2 to 3 hour incubation with our XTT
You should be able to see a gradient, but we did 3 hours, and so this may be maxing out. But we'll see under the plate reader what it says at 490 nanometer OD. So you're going to get a clean plate, and you're just going to transfer the supernatant. Okay, so it's going to be 75 to 80 microliters from each well into your new plate, and this is the plate that we're going to read, the one without the biofilms. And that's going to measure metabolic activity, which directly correlates with biofilm maturity. Okay, so I'm going to go from most, or I guess least metabolic activity to most metabolic activity. So it's going to be from lightest to darkest. So I'm going to start with the negative control, and I'm taking 75 microliters out. You'll see I must have gotten some drip from from row F into my negative control, as there's some metabolic activity there. So just something to think about. Okay. Now I'm going to start from column 1. About 75 microliters. And this is our positive control. Okay, you can get rid of your tips. And then you will make sure this is labeled, clean it off, and hand it out to your assistant for reading. Okay, so at this point, you have actually finished your XTT assays. You've read them at an optical density of 490 nanometers. And so you have all of your values with your plate. The last thing that you need to do is some sort of stat analysis. So what you will likely be doing is taking the average of your negative control. This is what we're going to call the background, and you're going to subtract that from all of your values. And so if you have a plate lid on, then your background's going to be higher. And so this is just to get rid of the kind of the noise that's already going to be in the machine, adding to your values due to the light diffracting through the like the plate lid and the plate base and all of that stuff. So once you correct for background, you're going to take all of your values and divide them by the average of your positive control, and that's going to give you your percent inhibition. So for instance, in lane 10, you might have close to 0% inhibition because it's so dilute, the compound that you were using, that it's going to look, very similar to your positive control. And so when you put the, bar graphs next to each other, you're going to see that there's not really any percent inhibition. Whereas, lane 1, maybe even lane 2, maybe even lane 3, depending on how effective your compound is, might have 100% inhibition. And so you'll be able to see, a kind of curve for each of the lanes. And then after that, you can use linear or nonlinear regression to estimate, at what value do we have 50% inhibition, at what value do we have 80% inhibition. Those seem to be kind of the standard of what you're using to measure minimum inhibitory concentrations. So then at that point, after you have enough data, you can say, this many milligrams per milliliter of heparin or doxycycline or whatever is the minimum inhibitory concentration of 48-hour mature Candida auris biofilms from this clade based on our data.
In addition to that, you may also want to test for statistical significance. There are a number of ways that you can do this. You can check our publications or our laboratory notebooks for more info on how to conduct that, and there's more than one way to do it. So thanks for joining me. I know it's been an incredibly long video, but I hope you've learned something, and please feel free to reach out to me if you have any questions. Thank you.